Hi, welcome everyone. Today we're going to talk about insulin and glucagon on glucose homeostasis. The pancreas is a heterocrine gland, meaning it has a dual function as an exocrine and an endocrine gland. Today we're focusing mainly on the endocrine role and how it regulates blood sugar or glucose homeostasis. There are tiny clusters of hormone-producing cells known as islets of Langerhans that create alpha cells, which secrete glucagon, as well as beta cells, which secrete insulin. A simple trick to remember this is that alpha cells secrete glucagon. They both have an A in their name. And think of the word glucagon, when glucose is gone. So the normal blood glucose range is about 80 to 120 milligrams per deal. When the glucose concentration is too high, aka hyperglycemia, insulin helps to lower it. And when the blood sugar drops too low, aka hypoglycemia, glucagon helps to raise it. When we eat a meal, glucose levels in the blood rise. This triggers a response in the pancreatic beta cells of the islets of Langerhans. Insulin production begins in the nucleus, where DNA is transcribed into mRNA. This mRNA travels to the ribosomes on the rough endoplasmic reticulum, where insulin is synthesized and processed. It is then transported into the Golgi apparatus for further modification and packaged into secretory vesicles ready for release. GLUT2 transporters allow glucose to enter the beta cells. Inside the cell, glucose undergoes glycolysis, generating ATP. Of course, there are many intermediate steps, including the link reaction, the Krebs cycle, and the electron transport chain. This increase in ATP causes potassium channels to close, leading to cell depolarization due to the positive membrane potential. Depolarization opens calcium channels, allowing calcium to flood into the cell. This calcium influx is the key signal for insulin secretion. Calcium triggers vesicles filled with insulin to fuse with the cell membrane, releasing insulin into the bloodstream to lower blood sugar levels. Insulin is released from the pancreas and travels through the bloodstream to the liver, where it binds to its tyrosine kinase receptor, a transmembrane signaling receptor. This activates the intracellular pathways, including the PI3K-AKT signaling cascade, which helps regulate glucose metabolism during glycogenesis. Once glucose enters the cell via GLUT2 transporters, it follows two main pathways: glycogenesis, where glucose is converted into glycogen for storage, and glycolysis, where glucose is broken down into pyruvate for energy production. Now, glycogenesis is the primary pathway under insulin signaling, ensuring excess glucose is stored instead of remaining in the bloodstream. Insulin is released from the pancreas and travels through the bloodstream to more target tissues, including muscle cells. Insulin binds to the TKR, the transmembrane signaling receptor, activating the same PI3K-AKT pathway inside the cell. This signaling promotes the translocation of GLUT4 transporters from intracellular vesicles to the plasma membrane, allowing glucose to enter the muscle cell. Unlike GLUT2 transporters in the liver, which is insulin independent and always present in the membrane, GLUT4 is insulin dependent, meaning it only moves to the surfaces when insulin is present. Once inside, glucose follows the same pathways, glycogenesis or glycolysis. Now, since muscle cells lack G6P, they cannot release glucose back into the bloodstream, ensuring that glucose is retained for muscle energy needs. The third and final target cell is the adipose tissue cells. Insulin binds to its tyrosine kinase receptor, the transmembrane signaling receptor again, activating the same PI3K-AKT pathway inside the cell. This signaling promotes the translocation of GLUT4 transporter to the plasma membrane, which, as we know, is glucose dependent. So again, unlike the GLUT2 in the liver, which is insulin independent, GLUT4 requires the presence of insulin. Once inside the adipocyte, glucose is primarily used for lipogenesis, the process of fat storage. With the PI3K pathway activated, glucose uptake increases. Glucose is converted into G3P, which then combines with fatty acids to form triglycerides. During fasting or a low blood sugar, the alpha cells of the pancreas release glucagon, remember, because glucose is gone. So glucagon will travel through the bloodstream to target tissues, primarily the liver, and also adipose tissue cells. Glucagon binds to the G protein coupled receptor, activating the G protein signaling pathway. This leads to the exchange of GDP for GTP, activating the G protein. Now, the activated G protein stimulates adenylate cyclase, which catalyzes the conversion of ATP into cyclic AMP, amplifying the glucagon signal. With cyclic AMP levels rising, protein kinase A is activated, triggering a cascade that promotes glycogenolysis, breaking down glycogen into glucose for release into the bloodstream. Remember, back when insulin was active, glycogen was stored just for this purpose. Glucagon also stimulates gluconeogenesis, producing glucose from non-carbohydrate sources that we will explore in just a second. Glucagon promotes lipolysis in adipose tissue cells by activating hormone-sensitive lipases, aka HSL. This breaks down the triglycerides into glycerol and free fatty acids, which can be used for energy. And as you can see, the glycerol travels back through the bloodstream into the liver and can be converted into glucose. This is similar for amino acids, which can also be converted into glucose and all released back into the bloodstream and successfully inducing glucose homeostasis.